So there are three main ways your body will defend you against these pathogens. It can be via phagocytosis, via complement-mediated lysis, and via cell-mediated immunity. Let's discuss them one by one. Well, the usual destiny of unsuccessful bacteria is death by phagocytosis. Survival, therefore, requires avoidance of this fate. Now, the main ways in which the bacteria in your body can achieve this lie in the capsule, affecting attachment, the cell wall, affecting digestion, and the release of exotoxins, which damage phagocytic and other cells. Talking more about phagocytosis, immune proteins like acute phase proteins, like complement and antibodies, bind to the surface of bacteria by a process called opsonization. Well, opsonization is the important process in your defense system, by which particles or complexes are made readily ingestible for uptake by phagocytic cells. In simpler words, specific serum proteins, known as an opsonin, coat the invaded bacteria. This opsonization allows the phagocytic cells, such as macrophages, to pick up the pathogen more easily and faster, and engulf them by the process of phagocytosis. Here, the most essential opsonin of all is complement C3b, which is a particularly important opsonization protein for controlling bacterial infections by this mechanism. Now, opsonization allows killing of gram-positive bacteria, like Staphylococcus specie, that are resistant to killing by membrane attack complex. After bacteria are ingested by phagocytosis, they are killed by various processes that occur inside the cell and broken into small fragments by enzymes. Phagocytes present the fragments on their surface via class II major histocompatibility, MHC class II molecules. Circulating helper T cells recognize these bacterial fragments and begin to produce proteins called cytokines. Two major groups of helper T cells are known as Th1 and Th2 cells. Now, you must be wondering, if both of them are producing cytokines, then where does the difference lie? Well, these cell types differ in the types of cytokines they secrete. Th1 cells predominantly produce interferon gamma, which promotes cell-mediated immune mechanisms, while Th2 cells produce mostly interleukin-4, or IL-4, which promotes humoral immunity by activating B cells. These B cells make antibodies that stick to extracellular bacteria and prevent their growth and survival. So this is the one way your body will fight the bacteria entering your body. Now, the other way your immune system can protect you is via the complement-mediated lysis. So when bacteria invade the body, they can be attacked by immune proteins called complement proteins. Well, the complement proteins assist in bacterial killing via three pathways: the classical complement pathway, the lectin pathway, or the alternative complement pathway, where the classical pathway is antibody dependent and thus part of the acquired immune response. Lectin and alternative pathways are part of the innate system, as they can be activated upon first exposure to a pathogen. Well, these all three pathways follow the same pattern once the C3 protein is activated. In other words, the pathways differ from one another in the way of their initial activation step, while the rest of the pathway is the same. We already had a detailed discussion on these three pathways in our video on the complement system, so here we are just going to skim through them for revision. Starting off with the classical pathway. So the first steps of the classical complement pathway require the binding of antibodies to the surface of the target bacteria. The antibodies then become targets for one particular complement protein complex, known as C1. Here, C1 binds to the tail, known as Fc region, of the antibody. Once bound, C1 initiates a cascade of cleavage and reforming of complement complexes that ends in the binding of several complement proteins to the surface of the bacterium in the form of a membrane attack complex, or MAC, or can generate opsonins that label a bacterium for destruction. Then membrane attack complex, or MAC, can insert into the cell membrane of gram-negative, but not gram-positive bacteria. There, it produces pores that allow the entry of membrane-damaging molecules, such as lysozyme, and makes the bacterium susceptible to osmotic lyses. Now, the second pathway, the alternative complement pathway, does not require antibody to initiate the lyses of bacteria. Here, complement proteins from a complex known as C3 directly bind to bacteria and activate downstream components in the complement cascade, once again ending in formation of membrane attack complex that causes lyses of the bacteria. Now, moving on to the lectin pathway. During the lectin pathway, mannan-binding lectin, or MBL, binds to proteins containing mannose residues that are found in some types of bacteria, such as Salmonella species. Once bound, MBL forms a complex with an enzyme called MBL-activated serine protease, or MASP. In this form, this enzyme activates C3 convertase by cleaving C2 and C4 complement components that participates in forming membrane attack complex, and the rest of the pathway is sane. Now, the third way to protect your body against invaded bacteria is via cell-mediated immunity. Some bacteria engulfed during phagocytosis avoid the killing mechanisms of the phagocyte to survive inside cells. Macrophages are a common target for intracellular bacteria like Salmonella species that live inside cell compartments. These bacteria cannot be detected by complement or antibody, but instead are eliminated using a cell-mediated response. Infected macrophages present bacterial peptides on their cell surface using MHC class II molecules. Well, this mechanism is called antigen presentation. Now, a helper T cell surveys MHC class II molecules with its T cell receptor, or TCR, to observe the peptides they hold. If a bacterial peptide is presented, the Th1 cell releases interferon gamma. This cytokine stimulates killing mechanisms, such as production of lysozyme, inside the infected macrophage to digest and destroy the invading bacterium. Interferon gamma also increases antigen presentation by cells, making the bacterium more visible to the immune system and hence more prone to attack. So these were the three ways your immune system can defend you against different bacteria that invade your body. Now, in the next parts of the video, we will discuss the various structures of bacteria and their role in generating an immune response. So let's watch the next section.